everyone. So now that we've talked a lot about the gross anatomy of the heart, understand the blood flow through the heart, and discuss the coronary arteries, it's time for us to start talking more specifically about the smaller structures of the heart so that we can better understand exactly how the heart is able to pump the blood. So what we're going to do is start by talking about the different cell types, and then from there we'll move on to drawing and understanding the conduction system so that we could relate some of the electrical function of the heart to actual contraction or the mechanical function. So the three types of cells that we're going to discuss today are pacemaker cells, conducting cells, and contracting cells. And later on, I'll mention some of the other names for these types of cells. So these aren't the only names, but these are the names that we're going to go by for now. I thought we would start by talking about the pacemaker cells. And the pacemaker cells are generally round in structure, and I just wanted to highlight a couple of points about them. One is the cell-to-cell connections, and the other is a few of the organelles. So this is not necessarily comprehensive, it's just a little something to put into perspective relative to some of the other cells that we've previously discussed. So these cells are connected to each other by two of the three types of cell junctions that we've discussed previously. The first is gap junctions, and the gap junctions, if you remember, are the channel proteins, the connexons, that allow for flow of ions directly between cells. So rather than, for example, in neurons where we talked mostly about the chemical synapse, we could think of this almost like an electrical synapse, but instead of talking about neurons, we're talking about a variation of cardiac cells. And so these gap junctions allow for the passage of ions between the cells in order for them to generate action potentials in subsequent cells. And we'll expand on that in another video, so it's going to be totally separate because it's actually not, there's quite a bit of information about it. Also, there are desmosomes that link the cells together, and if you remember, that's kind of like stitching the cells together so that they don't come apart. And we'll talk a little bit more about why that's important when we talk about contracting cells. The pacemaker cells also have an abundant amount of mitochondria. There's a pretty significant storage of glycogen, and they have an arrangement of these things called myofibrils, but the arrangement is a little haphazard, I suppose, compared with the organization that we'll see for some of the other cell types. So I just want to expand on that a little bit. So if you remember, there were four types of proteins that are found as part of the cytoskeleton. They were microfilaments, microtubules, intermediate filaments, and thick filaments. And the microfilaments are also known as actin, and we said the thick filaments are also known as myosin. So in muscle tissue, actin and myosin are really critical to performing the actual function of contraction. So for now, let's just think of myofibrils as these little structures that contain actin and myosin, and they're types of cytoskeleton, parts of cytoskeleton that are necessary for contraction. And so, as I said, the myofibrils are I guess, strewn about in these cells, but not necessarily organized in any like linear fashion. The next type of cell that we want to discuss is the conducting cell, and the conducting cells are more cylindrical in structure. They are a little bit on a wider side compared to the contracting cells, and they have some of the same important structures. So gap junctions, again, allow for the exchange of ions between cells. There's abundant mitochondria, and there is an array of myofibrils, but again, not necessarily in an organized fashion. And then lastly, the contracting cells. So the contracting cells are essential to allowing the heart to actually perform the pumping function. And these cells, as compared to the conducting cells, are a little bit more narrow, and they're longer. And they have this branched arrangement. So each one of the cells is marked its boundaries by the purple lines. And the cells are connected to each other by these things called intercalated discs. So intercalated discs are specialized junctions between cardiac contracting cells, and they're basically made up of gap junctions and desmosomes. So they're not as fancy as they sound, but again, those structures are really important. Gap junctions allow for the exchange of ions between cells, and desmosomes are essentially the stitching that holds the cells together, which will prevent, as these cells are contracting, they're expanding, relaxing, and we don't want them to come apart. And so the presence of desmosomes is essentially what ensures that the cells aren't going to separate during contraction. Contracting cells also have abundant mitochondria. And the other thing that I want to mention, and this hopefully is a little familiar, is the presence of striations. And so we mentioned that the types of tissue that are striated are skeletal muscle tissue and cardiac muscle tissue. And the striations arise because of a linear arrangement of actin and myosin. And I know that it's deceiving in this picture. If we were to consider the arrangement of actin and myosin in this drawing, it'd actually be in a vertical fashion. So it's not the same direction as the striations. The striations are kind of like stripes, but and they're seen particularly when you look at these cells under a microscope. But the arrangement of the actin and myosin allows for these striations to appear. So there's a lot more to say about the specifics on contracting cells, but when we talk specifically about the physiology of muscle contraction, we'll draw another picture of these cells and talk a little bit about them in more detail. So before I draw the conduction system specifically, I just want to introduce another principle, and this principle is automaticity. Automaticity means that cardiac muscle beats in the absence of neural and hormonal stimulation. So we've mentioned before that in terms of regulation of body systems, many times there's a local amount of regulation that occurs per body system, as well as a contribution by the nervous system and the endocrine system. But the other thing to keep in mind is that there are certain body functions or activities that are absolutely dependent upon either the nervous system or the endocrine system. A common example would be skeletal muscle is unable to contract in the absence of neural stimulation. So somatic motor neurons are going to deliver the action potentials to the skeletal muscle cells, and then that will generate the appropriate response in order to allow for muscle contraction to occur. It's important to distinguish cardiac muscle from these other types of organs and organ systems because it does not require any innervation or any function from either the nervous system or the endocrine system in order to pump the blood. And so I think to demonstrate that a little bit more clearly, we could cut the heart out of the body and it would still beat. And obviously once the delivery of oxygen ceases, it will cease to produce ATP and therefore it won't be able to beat anymore. So it's a time-sensitive event. But in terms of generating that stimulus to tell the heart to beat every single time, that's coming from an internal structure. It's not coming from either the nervous system or the endocrine system, like so many other types of organs and tissues depend on. So there's a general flow of events that occur, taking into consideration the cells that we've just discussed. First, the pacemaker cells will initiate electrical impulses. Second, the conducting cells will distribute these electrical impulses to the contracting cells. And third, the electrical impulses arriving at contracting cells will cause cardiac muscle contraction. And so as we talk more specifically about the conduction system, it will be helpful to keep this scheme in mind so that the process doesn't become too overwhelming. All right, so now that we've talked about the overall scheme of how the different types of cells may interact with each other, I'd like to be a little bit more specific about what the conduction system actually looks like and where the specific types of cells are located and what the various structures are responsible for. So let's just, we'll draw the heart again and take a look at the conduction system. So everything in red is essentially the conduction system, and we'll just kind of work our way down one step at a time, and I'll do it in a linear fashion, so the same order in which the communication would occur. So first we have the sinoatrial node, or SA node, most commonly referred to as SA node, which is the official pacemaker of the heart. So therefore it's comprised of mostly pacemaker cells and maybe some other conducting cells to communicate between the pacemaker cells or between the pacemaker cells and then the atrial cells that are found in both of the atria. So the SA node is located in the posterior wall of the right atrium, kind of near the entrance for the superior vena cava. and it has an inherent pacing rate of 80 to 100 beats per minute. And what's very interesting actually is we know that a normal adult heart rate is between 60 and 100, so thinking of the SA node beating faster than many people's heart rates actually are is kind of interesting, but actually it's parasympathetic innervation, parasympathetic tone that's going to drop that down, and we'll discuss the neural innervation in a later video. So once the SA node depolarizes, and we'll talk specifically about the action potentials that are going to come from these different types of cells, once the SA node depolarizes, it wants to essentially communicate with the atria, and then also with the AV node, which we'll talk about in a second. So there are three essentially internodal pathways that connect to the next node, as well as this other structure called Bachmann's bundle, which connects the SA node to the left atrium. And it's a little bit controversial whether or not there are truly distinct pathways made of conduction cells along which the impulses would be conducted, but it's, in some textbooks it's very clearly described. In other textbooks it really indicates that there isn't much, and that the conduction may occur just between atrial cells themselves. So in either case, whether or not there are truly there's truly a specific set of conduction pathways, or maybe just a random array of conduction cells, or not even, the expectation is that the SA node is going to initiate the electrical impulse, and then those pathways, or the cells of the atria, are going to then transfer that electrical impulse to the AV node, so that the lower half of the heart, the ventricles, will be able to receive the impulse and also contract. And then also the impulse is going to spread to all of the atrial cells, so that the atrial cells will become depolarized and able to contract as well. Now, there are also some ectopic pacemakers that are thought to be present in very close proximity to the SA node, and if there are internal pathways, then they would be along the internal pathways specifically. And these ectopic pacemakers have an inherent pacing rate of 60 to 80 beats per minute. So in a normal circumstance, the SA node is doing the pacing, and because it's pacing at a faster rate than 60 to 80, the ectopic pacemakers generally won't be able to provide any particular direction to the rest of the heart. But if the SA node was to fail for a variety of reasons, then possibly the inherent pacers of the atria would be able to take over. So as we mentioned, the communication is seeking to arrive at the AV node. So the AV node is the atrioventricular node, and it's actually comprised of fewer pacemaker cells than the SA node, and also some conducting cells. It's located in the floor of the right atrium, towards the interatrial septum, and it actually functions as the only point of connection between the atria and the ventricles in terms of electrical conduction. So the valves, and there's a layer of connective tissue that basically electrically insulates the atria from the ventricles and keeps them totally isolated, and so the only way for them to communicate with each other is through this single pathway. So the cells, especially the conducting cells present in the AV node, have fewer and more dispersed gap junctions compared with other conducting cells, particularly in the ventricular pathways that are coming up. And the AV node is more narrow compared with the ventricular pathways as well. And so these two sort of adaptations allow for very slow conduction through the AV node. So the conduction basically slows down. And that's important because during the time where the conduction is slowly passing through the AV node, we also are giving the atria time to actually contract and push the blood into the ventricles. The AV node is contained within the AV junction, and the inherent pacing rate of the AV junction, largely contributed by the AV nodal cells or pacemaker cells, is its pacing rate of about 40 to 60 beats per minute. Now, once the electrical stimulus passes through the AV junction, it arrives at a couple of structures. The His bundle or bundle of His followed by the bundle branches. So it splits into two bundle branches. And actually, although not depicted in this drawing, the left bundle branch actually has an anterior and a posterior fascicle. So there's actually two parts to the left bundle branch, and we'll talk about that more later. For now, we'll just draw it in a simple fashion and indicate that there are two bundle branches, right and left. And then the bundle branches, as they make their way down on either side of the interventricular septum, they'll reach sort of the apex of the heart and then start to work their way back up along the walls. And the cells that make up this entire structure are referred to as Purkinje cells, also conducting cells. So in some cases, the conducting cells could also be referred to as Purkinje cells. And there are, once the bundle branches reach their kind of final endpoint, they will branch off into what's considered the terminal filaments of the Purkinje fibers. Now, compared with the AV node, these pathways are wider, and they are made largely of conducting cells, and they have a few ectopic pacemakers along the conduction pathway as well. The primary function is to distribute electrical impulses to the ventricles, and the conduction through these pathways are extremely fastest out of the entire heart. And the ectopic pacemakers, should the pacemakers above it fail, have an inherent pacing rate of 20 to 40 beats per minute. All right, so we've talked about the different types of cells that can be found in the heart. We've talked about the overall scheme in terms of how electrical impulses have the ability to affect contraction, and then we've discussed the basic components of the conduction system. So there's clearly a lot more to discuss. We have to talk about the different types of action potential as well as the physiology of muscle contraction in order to get a much more clear understanding of how the heart is working. So we'll be covering those in the next couple of videos. But that's all for now. Thanks for watching.